to call your attention to a conference that we will organize from 7th to 8th of November of this year in the University of São Paulo in honor of Professor Iberê Caldas, who is completing 70 years old, and many people who worked with him, collaborators, friends, and this invitation is being extended to all of you who would like to contribute. So thank you very much, and let's...
Okay, thank you, Ricardo. Our first speaker.
Can you hear me? Yeah?
Yeah. Kelly Iarosz, and she's going to talk about mathematical model of brain tumor.
Tá.
Para poder ligar aqui. Obrigado, pode contar.
Escolha que segue. Good afternoon, everyone. Thanks the organizer, and thanks everyone for staying here after lunch. Today, I'm talking about the mathematical model of brain tumor. For some person here, maybe this is strange because sometimes, did you see I talk about the plasticity? But during my talking, I connected this, and then you can understand why I talk about this model today. Here I put some names of the collaboration, but this collaboration is not just one collaboration. We have collaboration with these persons, these researchers. Started this in 2009. I am part of the Professor Iberê Caldas group in São Paulo University at the Institute of Physics, and the part of the 105 group size. Here we have some persons of this group, and in my last slide I pay attention of the names of the persons goes to the poster presentation and another speakers. And then properly I talk now about my talk. Sometimes we present about the brain and talk about the plasticity and different topologies and some neuronal models. Or we talk about the cardiac considerations, but today I'm talking about the lesions. The cancer or the tumors are maybe the main and more aggressive form of the lesions in our brain. And then we have some considerations about our cells. In some cases, we have a cell mother, and then this cell reproduced, separated. During this separated, we have a cycle cells, and after the separation, we have a new two copies. But these normal cells can be divided around 50 times, one normal cells. When these cells suffer one deformity in DNA, the cells can be separated more times. But when this occurs, we have some deformations. And this deformation sometimes is called the cancer or tumor. We can separate this in begin, or we can separate it in malignant cancer or tumor. But the main case, or the main consideration in this talking, is we have the cell division. Our cell division is not normal. We have one damaging in this, and then we have a formation of the cancer or the tumor malignant. Here we have one short video about the metastasis proliferation. In this case, we have just compression of the neurons. In our case, we don't use just the compression. We need to analyze what's happening with our neurons. And then this case of the metastasis is just one example from the lung to the brain. My talk is probably about this case. We have the glioma times the neurons. Why this? Some studies clarify the glia cells and they call in the name of the babysitter of the neurons, because the neurons have no quality for providing nutrients or care for alone. And then, who cares for the neurons? Glia cells. Glia cells are one class of the cells, and these cells can divide in another. But the main responsibility of these glia cells is care for our neurons, provide the nutrients, regulated our system, and what is important in this. I pay attention to this because if you have neurons, and here we have neurons, and then we have glia cells. These glia cells supported our neurons. Neurons cannot live alone. And then, if your glia cells death, your neurons death. If you lost this support, our brain have some damage. And then, here we have one example of the proper case. In this case, we have one cut of the brain. And I know this picture is not so good, and the main now, because we lunch now. And then we have one cut. And this cut, we can look the proliferation of these glia cells. After this proliferation, some neurons death, and the part of our brain death, properly apoptosis, or necrosis, and some specific name for this. And then we have a part of our brain lose. We can discuss this in terms of the plasticity. We can conduct other discussions about the topology. But the main case is this lesion, this problem, this disease, case one damage in our brain. I can analyze the signals after this, or I can analyze what's happened with our new connections in this brain. But first, we need to know what's happening with the neurons and what's happening with our glioma, or glia cells proliferation, and we have no treatment about this. Just put in this case and look for the ground. No, we have treatment. But the treatment is not so, the health is so soft. And then some perturbation in the system causes some effects. We have one specific form of the treatment, but not just this. We have another forms of the treatment. I pay attention to this, but we have more. Why I can pay attention to the chemotherapy? First, because the model is about the chemotherapy, and then first because of this. But the system of the health in Brazil offer sometimes this type of the treatment. And then what's the protocol for this treatment, this type of treatment? The person goes to the hospital or specialized clinics, and then wait one doctor and wait the protocol for drugs, for receive these drugs. And then the agent chemotherapy is infusion in the person, and then the person wait. This, for us, we put some parameters in our equations, evaluated this in our computers, and they wait for our beautiful pictures. And then goes here and present some graphics. But the persons with the weight, these persons, it's calling them the so rude, so aggressive time for your life. And then some aspects are considered during this wait. Main, the hair loss. This is an effect more visible because the persons some persons, when you look for the talking, the persons have a cancer. These persons have no hair, or sometimes these persons have the ability of the face, the health. And this is because the infusion of the chemotherapy. The chemotherapy agent is aggressive for all the cells. Remember, I talked about if you have neurons, you have glia cells. These glia cells can be proliferation, and then we have one specific type of the cancer, called the glioma. The glioma can be attacked from the chemotherapy agents, but not just our glia cells. When you receive the chemotherapy agents, you receive attacked in your normal cells, in your glia cells, and your neuron cells. Because of this, we study during some years any considerations in cancer models. I would like to inform this. This is like my first love in science, because when I started my PhD, I studied this model. This is our first model I studied. And then in 2010, we studied about the suppression of the cancer, and then we can insert the delay in cells proliferation for consider the mutation. After this, you can insert the immune system and try to study what's happening with the immune system fortaleced, because this is one type of the treatment. And then we study what's happening if we use the chemotherapy agents in pulsed infusion and continuous infusion, because we have these two types of the infusion of the chemotherapy. And then we study what's happening with the glia, neurons, and chemotherapy agents. After this, we study what's the relation between the dose-dense chemotherapy, because this is influenced so much. And then we study one phenomenon, clinical phenomenon, calling the Norton-Simon hypothesis. And after this, we study the rules of this dose-dense chemotherapy. And this paper, this is our last paper submitted, it studied the blood barrier in our brain. I call in pay attention to this because the José Trobia, where is the José Trobia, there one poster presentation about this paper. Then, okay, and then you can see in this timeline, we have some considerations about the mathematical models and the connection between the glia, neurons, and chemotherapy. Basically, our model show the neurons and the influence directed the glia cells in neurons. We have the competition between the glia cells and the cancer because the glia cells and the cancer is connected. And then we have the influence directly in neurons, in glia cells, and the cancer cells when you put in your body the chemotherapy agents. This four equation represents our first consideration of the models. We have the first equation, and consider in this first equation the glia cells concentration. In second equation, we have a representation of the glioma cells. In this third equation, we have the neurons concentration in our brain. In the last equation, we have the concentration of the chemotherapy agent. I would like to show the interaction coefficient are so influent in our model. The proliferation rate are influent because we have if you put the no realistic values in this model, we have no brain. We can insert some values and the simulated proliferation for all life. But if you don't limitate, you have one volume, a big volume. And in our brain, we have no big volume. We need to limitate this. And then we have some consideration about the chemotherapy agents, because we try in our model the realistic drugs of the pharmaceutical industry. And then we try to use the drugs of the public system. And in my final slides, I show for you some considerations, because we receive this treatment, but this is not the best treatment today. But the public system offers this, and then you need to study what this public can be offered. Our results show the main results. I would like to pay attention to this. Treat one tumor is better than non-treat. This is not discutable. In this case, we have here one example. If there is no treatment, you don't insert any infusion of the chemotherapy agents in your treatment. Just wait the proliferation. And then when this occurs, the glioma grows, and then your glia cells down, because the glioma took the volume of your brain, and in consequently this, the neurons death. Remember, if you have no support, your neurons death. And then, if you try to apply the chemotherapy, for some time you receive the chemotherapy, the form continues, and then you can observe what's happening in your model. We have one evolution about the glia cells. We have the reduction of the glioma cells, and you have no neurogenesis of the neurons. And then you can remember the chemotherapy agents don't attack just the glia cells and just the glioma, attack to the neurons. But this loss of the neurons is not so aggressive like don't receive the treatment. And then we have here some considerations about remember about the volume of the brain. You need to put one limited of this, and then this is a carry capacity. It's like a density of our brain. Mass and volume, the reason about this. And then we have this constant, but it is represented, this alpha, represented the influence, how the neurons you lose inside of this volume, considering the carry capacity. And when you look at this, we can observe the best of the consideration of the treatment about the chemotherapy. In this case, we have here the color bar, and look the concentration of the neurons. In this case, in yellow, we have a reduction about the 2% of the neurons. And in this case, we have around 70% of the reduction of the neurons. In this green line, we have a mark of the alpha around the 2. And here we have the chemotherapy around the 16. Here is normalized model, but in real life, this is equivalent and 75 milligrams per liters of the infusion in your body. In this case, continuous. Imagine one person goes to the hospital, stay in the hospital, and then receive every day one infusion of the 75 milligrams for milliliters of the chemotherapy agents. And it is added today, and after this person receive, and after receive during the 1 month, for example. And the case of the health of the person decrease a lot. Because the cells of the body death, all the cells, the concentration of the cells death. And then, because of this, we consider this time space of the study. But I think the main results about this stay, the figure 5 compared of these two lines. About time, because we have one first work and they explain about the suppression of the chemotherapy, the cancer, when you put one high concentration of chemotherapy or one low concentration. But the main study about this last figure stay here. When you infusion the chemotherapy and this is continuous, you have so damage in your body. You can kill the cancer cells, but you kill your body too. And then if you consider not just the infusion continuous, but you can consider one dose dense around this is the limitation of the pharmaco, this value, but during just 5 days. And then stop for 23 days, and the body recover your health, and then this body can be receive the drug now. But in this case, we have the regression of the glioma cells. We have one recover of the glia cells, and you have stabilized the neurons loss. But no, this type of treatment is not offered. Sometimes is not offered by public treatments. And then, because of this, I consider the last concluded reflections and pay attention of this conclusion about it. We have one model. We study about the type of insert chemotherapy. We know about the difference when you try the pulsed chemotherapy or the continuous. We know the main drug used in the normal treatments, but we know this is not the best. We have another types of the drugs, but it's not offered. This is just types of the offer. I remember this type of cancer is glioma cancer, and then is a specific drug for glioma. If you have interest in this and know more about why and about the carcinogenesis, you can read more about this in this paper. When you put in Web of Science, for example, this is one of the main papers about the explanations, because the cancer is not just the consideration about the external factors. And this is one attention. I remember, be careful with the news in our main journals like Facebook, because sometimes the news is not real. This is one example. This is permanecing the network during days. And some persons talk about the tumor necrosis factor in banana. This is crazy. If one person discover this, and then we can go to the Nobel for this person, because necrosis is factor of the human cells or the animal cells in some considerations, not the vegetative. And then, but this is a main information. The news in some network is banana has tumor necrosis factor. Eat banana like this with the spot, the black spots, because this is important, black spots. And then you can cure the cancer. But the original news is not this. It's one injection of the one specific drugs in rats, in mice, can be observed once some necrosis of the tumor in mouse, not in banana. And the banana is involved in extracted from banana. And then be careful with the news about the cancer and the same other diseases. Thank you very much. I would like to invite you to visit our website. And I would like to invite you to visit the Trobia poster, the José Trobia poster, the Paulo poster, Paulo Protashevsky, and tomorrow we have the talk with the Fabiano Ferrari. It's this. Okay, thank you.
Thank you for the nice talk. Questions? Comments? Ram?
Yeah. I was just wondering, how far are you from having clinical trials and the possibility of implementing some of these ideas?
And then we try the drugs and the actual protocols in the system of the health consider the pulsed chemotherapy. Yeah. And then but this is around 5 years ago. Just 5 years ago we started this. And the pharmaceutical industry use this some consideration. We have so many citations in our paper from 2050 doctors in medicine, in specific treatments of the medicine.
Trying it?
Yes, the community try to apply our model in your types of treatments. I wait, try more, but I wait.
When you do these trials, as you're just answering, would you try to apply this model? It would be in the way the chemicals are used. I mean, do you need permission from Anvisa?
We have no tests in humans or rats or some cases. But if one doctor, one medical case goes to apply, and then he needs, yeah. But we don't need it because we take the data from another papers or the drugs tested from industry. Yeah. We try to use this type of the drugs for test our model first. And then we decide use one or two or three specific drugs. Yeah. Okay.
Thank you very much.
Thank you.
The next talk is not here, so we move to the following talk, and then we'll have a longer break for coffee. So the next speaker is Alexander. Alexander? Yeah. And he's going to talk about properties of liquid water from atomistic simulations and neural networks.
In the ribbon side. Yes.
Me sorprendía que no hacía ninguna pregunta. Estaba inquieta. Me dejas ahora más tranquila. Porque hiciste una pregunta. Yo decía, ¿qué le pasa a esta que está tan